you. I think we have these microphones on. Can everyone hear us? Yeah, okay, there. So let's talk about microbes. I love microbes.
Oh, well, we were just chatting, and I mentioned that, well, the endocannabinoid system, to a certain extent, particularly through the CB2 receptor, is a protective system. Now, one of the things that we have to protect ourselves is tolerance of microbes to antibiotics. Now, what does that mean? I mean, our body and microbes have been talking to each other for millions of years. And chances are that the body attacks microbes, or at least certain microbes, and microbes respond by taking protective measures. And then the body tries to find ways to overcome the protective measures, and it goes back and forth. Now, as obviously, at the moment, one of the major problems in medicine is what do we do with this tolerance of microbes to antibiotics? If things do not change, and seriously change, within 10 years, we may not have any real antibiotics around, a situation which is extremely dangerous. But we do have the endocannabinoid system, so we thought, let's see whether the endocannabinoid system is somewhat involved in that kind of conversation between microbes and humans. So I have something on the Fireside chat which is not immediate. It comes somewhere. Oops, here it is. Can we go one back?
Oh, the red one will go back.
The red one.
And the laser is the green one. There we are.
Yeah. Now, we speak to each other by making noise. We call it talking.
Chatting.
Chatting, if you wish. Microbes talk to each other by releasing chemicals. And when microbes are in stress, they start excreting chemicals, telling other microbes, not one or two, but millions of them, let's do something. And this is called quorum sensing. Why called quorum sensing, I have no idea, but that's what it's called. And one of the ways they protect themselves, they come together and form a biofilm. So the quorum sensing, as you can read, is the ability of bacterial population to chemically communicate with each other. And one of the ways bacteria protect themselves from stress, and antibiotics for them obviously stress, they make biofilms. Biofilms of millions of microbes. The antibiotics cannot go in. In the biofilm, the microbes do not develop, and if they do not develop, then the antibiotics anyway do not act on them, so on. And they sit there and wait until they can do something. So this is one of the ways microbes protect themselves against the stress of antibiotics. One way of tolerance, if you wish. Now, we thought if we have an endocannabinoid system, which is a protective system, does it do something? And my colleague, one of my colleagues who knows how to do these things, I haven't got the foggiest idea how to work with these things. We gave him an extremely potent synthetic cannabinoid, which is very potent both on CB1 and CB2. We thought, well, maybe we shall take something very potent, see whether it acts, and then we go to CBD, THC, endocannabinoids, and so on. This is a compound called HU210, which is about 100 times more active than THC, and wanted to see whether it affects the microbes. And yes, we reduced the formation of biofilms. It also reduced the formation of virulence factor production by these particular microbes. So we are going on, and with other microbes. We're going on in other forms of protection, but it seems that the endocannabinoid system, at least this is the first evidence, I hope this is not the last evidence, that the cannabinoid system interferes with the signal transduction used by bacteria, by the protection of bacteria against stress, which means that maybe, just maybe, in a couple of years, antibiotics will be administered together with endocannabinoids or particular endocannabinoid to reduce the tolerance of bacteria to antibiotics. So this is an answer. Shall we be there someday? I sincerely hope so. One of the ways to go ahead. Maybe it's a mistake. Let's find out in 3 years' time.
So just before we jump on to the next topic, I find it interesting that there are some class of antibiotics known as aminoglycosides that act specifically on the mitochondria, and we even have some patients that when there's a mutation in the mitochondria, the aminoglycosides can actually induce a form of hearing loss because of these. So is it possible that the cannabinoids are working through a different mechanism for their quorum sensing or dissolving of quorum sensing, or what do we know about its mode of action in the microbes?
Well, quite frankly, we don't know anything at the moment. This is the first evidence we have. I'm talking to the microbiologists, and they have never heard of endocannabinoids. They did normally, microbiologists don't know of the existence of this thing. And they look at me and say, oh, really? And, but it makes sense that our body has a defense mechanism against something that is normally occurring. Bacteria sense that the body is fighting against them. In this case, it's a synthetic compound or semi-synthetic compound that we give, and it's attacking them. But they don't seem to know that. They know that the body is attacking them, and this is their reaction. Making a biofilm, making other things. Making a biofilm to protect themselves against antibiotics. We have to learn a huge amount of things.
Right. It could be cytokine-based. You do tend to get these cytokine storms when there's inflammation, and it seems as if the cannabinoids are having some role in dampening down that inflammation. There's, I think, some talks today on PANS and PANDAS where we see this happening in pediatric patients. But yeah, it's fascinating. A lot of work to do. Maybe a lot more sequencing.
Well, we know that cannabinoids, some cannabinoids, endocannabinoids most definitely act on cytokines. This type, another type, many of the cytokines. We published quite a bit on that, and most definitely they act on the cytokines. How relevant that is to
quorum sensing.
What I was telling you about, I haven't got the foggiest idea.
Interesting. Well, on the sense of bone remodeling, our mother recently broke her leg in 6 places, and so we've been paying a lot of attention to your work here in osteoclast and osteoblast development. Tell us more. Well,
this started from a completely different research we were doing. It had nothing to do with cannabinoids. There were papers and statistics that women, after the age of 50, when there are hormonal changes, women have much less osteoporosis in the Mediterranean region than, let's say, Norway. And the reason for that, people said, was olive oil. We thought, well, olive oil, all of them use only olive oil. So we thought, okay, let's look at the oleic acid, which is the principle of olive oil, nothing to do with cannabinoids at this point. And so we looked at olive oil, and my friend Itai Bab, who is a specialist in bones, didn't find anything. So we didn't throw it away. They said, maybe it's a metabolite of oleic acid. So we looked at what kind of metabolites these acids make, and one of them, we thought maybe, for all kinds of reasons, I wouldn't go into that, fatty acid bound to amino acid. And we made oleic acid bound to serine, an amino acid. Now, this now brings us closer to endocannabinoids, because the endogenous cannabinoids are compounds of that type. Fatty acid bound to an amino acid or a derivative of amino acid. So we are now closer, all of a sudden, to endocannabinoids. We want to go away from endocannabinoids. So we looked at this particular compound, oleucirine or OS, and we found that it is present in bone, and Itai found that it is extremely potent against osteoporosis. And so why did we look at oleucirine? Well, those of you who remember your chemistry, this is anandamide, and there is a oleucirine on the top, and arachidonoylserine, which is close to anandamide, etc., etc. And at the end, I mean, following this particular line of thought, we have the oleucirine. This was published 5 years ago in Proceedings of National Academy of Sciences. And this particular compound, oleucirine, turned out to be extremely potent in bones. It enhances the activity of cells that produce bones, and and here we have, you can see going up and down, and it lowers the activity of the cells that destroy bones. Okay, that's fine, but does it work in osteoporosis? So we took poor mice, we took away the reproductive system, which essentially is what is happening in women after the age of 50, not exactly, but more or less. So they don't have the hormones, and these mice, after a few months, develop osteoporosis like women after the age of 50. So this, if you look at the red on the slide, you'll see that the bones, bone volume goes down. At this point, we started giving them oleucirine, and lo and behold, we saw now that it stops the reaction, it also moves it up, so the bones start being formed again. So here we have an endocannabinoid-like compound which works in a direction which we couldn't even think about at the beginning. Now, this is something that we have to take into account. We have about 150 compounds of this type in our body, in our brain. So in addition to anandamide, which acts on the endocannabinoid system, we have an additional 150 compounds or even more of this type, endocannabinoid-like compounds, and a few of those only, very few, have been looked into. So we can expect a huge amount of things happening, of which at the moment I can only guess what they are. One speculation, if you wish, has to do with psychology. I went to some of our psychology and asked them, what do we know about the biochemistry of individual differences? Of why am I different than anybody else here? Here we have a couple hundred people. Each one of us is different. Why are we different? What is the chemistry and the Nobel? And they said, we haven't got the foggiest idea. Well, is it possible, then I said, that because we have 150 or maybe 200 compounds of this type, endocannabinoid-like compounds, those that we have looked into seem to act on the central nervous system. Is it possible that the relations of these 150 compounds have something to do with our personality? Well, he said, I have nothing against it. So maybe our personality, to a certain extent, is based genetically on the 150 to 200 anandamide-like compounds that we have in our brain, and the relations between them will show someday that they are the basis or one of the bases of personality. Complete speculation. Somebody comes up and says, all that is silly, I'll accept it.
So if somebody tells me that I'm hard-headed, it's because these things are affecting my brain and the bones around my brain, right? And I'm excellent.
Well, would you like me to tell another short story?
Yes.
Of something that this is the, why do cannabinoids act on so many things? Well, we really don't know. One possibility is they have, or at least cannabidiol has, anandamide has, the ability to act through epigenetics. What is epigenetics? It is the effect on DNA without breaking it, just modifying the activity of DNA by methylating here, let's say, a part of the DNA that's involved in a certain reaction and causes the production of peptides or proteins somewhere. And works over time, and this working over time causes a reaction, causes a disease. If we block the activity, it can lower it. Well, it was shown by an Italian group that cannabidiol and anandamide can do that in a particular skin disease, in psoriasis. If this is a general reaction, then it may explain why cannabinoids have such a wide type of reaction. Maybe, just maybe, cannabinoids, not only in that particular skin reaction, but maybe in other diseases causes changes in the DNA epigenetics. Who knows? Again, it's speculation. You were asking me, where are we going? Well, here are some speculations. Some of them may be right. Some of them may be completely wrong.
There's an interesting model for memory in a model organism called aplasia, and we did some sequencing in this before because it's the largest single cell neuron, and the genome copy number in this is like 100,000 throughout the cell. But in sequencing it, there's a very interesting connection with 5-HT, which is serotonin, and methylation, and we could actually sample different areas of this single cell when it was trying to form, we believe, memories, and see different patterns of methylation and RNA moving around at a single cell level, which is really fascinating if these govern everything in there. I don't know if there's an endocannabinoid system in aplasia, but I'm going to go home and look.
Well, if we're speaking of endocannabinoids or not, I mentioned several things in my lecture, just passing on. One of them is we need clinical trials. We've talked about that for quite a few minutes. Again, I repeat it, we need clinical trials. We can't go ahead without that. A second thing is we need to look at the entourage effect. We published on the entourage effect 20 years ago. There has been no research on it. We should do. Many other people should work on the entourage effect. THC is not the same as cannabis with some other compounds there. What are we looking at? We don't know. We don't know whether it's THC with CBD is better for what disease, at what concentrations. We heard from the clinicians that, yes, we need more CBD, less CBD, depends on the disease, depends on the age, and so on. We know so very little. And the third thing that we have, there are many other things that we have to know, but another thing that we have to know is what are these endogenous-like cannabinoids doing? I mentioned now the possibility that they have to do with protection against microbes. There are many other things, but let me end on a positive note. There is there are lysozymes which are present in many animals. These are cellular organelles that have function by digestion and recycling of macromolecules, proteins, and so on. Fine. Now, somebody looked at the lysozymes in a worm. There is a worm called C. elegans. Now, people that work on longevity work on C. elegans. Why? Because the worm, C. elegans, lives only a few months, I think, and it's easy to work on it. I mean, working with a human being on longevity, well, we don't have the time to do that. But working on a worm that lives just a very short time, that's a good idea. So almost all people that work on longevity work on the worm C. elegans. And in a very recent paper, just a few months ago, it was found that this part of the cell, the lysozyme, takes proteins, and one of the compounds that under certain condition it can make is an endocannabinoid-like compound called olehyl ethanolamide. You may remember I just was speaking of olehyl serine that has to do with bones. Here, the worm C. elegans makes olehyl ethanolamide, which is again is an endocannabinoid-like compound. And lo and behold, this endocannabinoid-like compound, through a certain mechanism that they found, this was published in Science, activates certain genes and activates the transcription of genes of NH80 and promotes longevity. So the next idea is, well, maybe as of tomorrow morning, each one of us should take a spoonlet of olehyl ethanolamide and live to be 120 years old.
Excellent. Yeah, and some worms, right?
Well, this is, it is a beyond speculation. This is just to end on a happy note.
Excellent, yes. All right. Well, with all of that, we would like to turn some time here to present Dr. Mechoulam with a Lifetime Achievement Award. This is our sign of appreciation for the cascade and the avalanche that he's created. Now, there's something attached to this award that's not very transparent or apparent from what Brendan's holding. This award is actually represents a 1 trillion base pair award. So we are going to offer up a trillion bases, I say that with a pinky by my thumb, or a pinky by my mouth, to Dr. Mechoulam to sequence whatever he wants to sort of better expand this space and to understand whether it's gene expression or methylation, what have you.
Thank you. Thank you very much indeed.
Thank you indeed. I'm so very thrilled to do this.
Congratulations, Dr.
Yes.
Yeah.
Sure. You're in? I'll come around the other side here. This is 1 million megabases or 1,000 gigabases. And we encourage everybody to tweet or email their ideas. We by no means understand all of this. We want to crowdsource the best solution here.
Thank you.
Thank you.